Hello everyone. I thought I'd continue the exam paper walkthroughs that I've been doing. Even though they might not be immediately useful to you, I think it's handy to have them on the channel, banked up, just in case you need them, and when it ramps up ready for exam season, all the way in the distant future when coronavirus hopefully won't affect it. So today's paper that we're looking at is the 2018 Biology Paper 1. So, I recommend either downloading the paper or printing out the paper and doing it along with me. So then you can see how I'd answer it, and you can amend any answers that you've made. So let's get going. So figure 1 shows one type of white blood cell, and you can see it's labelled here. What is structure A? Well, white blood cell we know is an animal cell because we are animals, okay? We are not plants. So we're going to be straight away thinking that it's going to be something animal cell-like. We've got four options. We've got cell membrane, cell wall, cytoplasm and nucleus. Now A is pointing to the outside, so that to me means it's either going to be cell membrane or cell wall. Because we know it's an animal cell, that means that it can't be a cell wall, because remember, cell walls are only in plants. Okay, so it can't be the cell wall, so that means it must be the cell membrane. White blood cells help to defend the body against pathogens. How do the white blood cells do this? Tick 3 boxes. Okay, this is really, really common throughout this topic, is to know the three things that white blood cells do. So they engulf pathogens. We definitely don't want them to clone pathogens because that means they'll make more of them. So they're going to engulf and digest them. They don't produce antibiotics, remember that's what we as humans have made and we take as tablets or injections or what have you. They produce something called antibodies, and you'll probably see this in the news at the moment when they're talking about antibody tests for coronavirus. So you're seeing if you've had coronavirus and if you've produced, your white blood cells have sensed it and done something against it. The other thing which they do is produce antitoxins. They definitely don't produce toxins because that would be not that nice for you. Antitoxins are things which help counteract the toxins which bacteria or other pathogens might make, and those toxins what actually makes you ill. So if you produce antitoxins, then that negates those ill effects. Looking at this graph then, measles is a serious disease. That's a nice understatement. A person can die from measles. Figure 2 shows the number of cases of measles in England and Wales between 2012 and 2015. Actually, if it carried on to today, it'd be up here again, thanks to anti-vaxxers. Use figure 2 to calculate the decrease in the number of cases of measles between 2012 and 2015. So, really key with these graph questions to use a ruler, and then you can measure more accurately. You should take a ruler into the exam, don't be silly and not. So I'm looking between 2012 and 2015, so I need to know what the cases were in 2012 and what the cases were in 2015. If I use that ruler and draw a cross, I can see here that it is one little square up between 2000 and 2500. So I need to know what the scale is here clearly. And if I've got 10 little squares between 2000 and 2500, that means that each little square is going to be 50. So if I've got one more little square, that means in 2012 I have 2050 cases. For 2015, okay, I can draw a cross again. Okay, I mean I could even see here, but I'm just doing overkill. But it's only it's 2 squares up, so that means that in 2015 there were 100 cases. So that's all well and good. We now need to calculate the decrease. So if I started on 2050 and I am now on 100, that means that I have decreased by 1,950 cases. So the answer is 1,950 cases. So just one reason for the decrease in the cases of measles between 2012 and 2015. The big thing here when you see measles is it's one of the M's in MMR. Okay, MMR is a vaccine which protects against 3 diseases: measles, mumps, and rubella. And cases about vaccines will often focus on MMR because there was a scare in the 1990s after a paper was published by Andrew Wakefield, who said that vaccines cause autism. And this was completely refuted, but that means that we had a delay and there was a reduction in people taking up the MMR vaccine. That went up, and we see the concurrent reduction in cases. So one reason for the decrease in number of cases, I would suggest, is more people were being vaccinated. Against measles. Okay. Turn over to continue the question then. Antibiotics cannot be used to treat measles. Suggest why. Okay, so this is important to think about what antibiotics treat. Antibiotics are good against bacteria. Okay, they do not work against anything else. The 4 types of pathogens that you need to know about at GCSE, and indeed I suppose are the 4 types of pathogens. There is actually one more, I suppose, called prions. The 4 types of pathogens you need to know about are bacteria, viruses, very current at the moment, fungi, so things like athlete's foot, and protozoa, or protist is another way of saying it. The key protist disease you need to worry about is malaria. Measles is a virus. So antibiotics only work against bacteria, they don't work against these. So the reason that measles can't be treated by antibiotics is because it is caused by a virus. Okay, so measles is caused by a virus. Antibiotics only work against bacterial disease, or against bacteria. Okay, gonorrhea is a disease caused by bacterium. Gonorrhea can be treated with antibiotics. Give one other way to control the spread of gonorrhea. Okay, gonorrhea is a sexually transmitted disease. Okay, so that's the way it's spread. So we need to think of a way where we can stop the spread of a sexually transmitted disease. Okay, so using condoms is a good way, as it is sexually transmitted. Okay, a scientist investigated how effective different antibiotics were at killing gonorrhea bacteria. This is the method used. They grow gonorrhea or bacteria on an agar in a petri dish. So here's the petri dish, and the grey is the agar. They place 1 paper disc soaked in water onto the agar. They place 4 other paper discs, each soaked in a different antibiotic. They use the same paper disc and the same concentration of these antibiotics, and they put it to incubate for 3 days. So you'll see the bacteria grow, and you can see these areas around the discs where there's no bacterial growth. So that means the antibiotic has gone into the agar and killed any bacteria and stopped the growth in these areas. Give one control variable the scientist used. So a control variable is something that you keep the same. Very often we talk about this as things which make a fair test, and that's kind of gone out of favour recently, but I think that it's really easy to remember it as the fair test variables. So one thing which I want to keep the same is they incubated it for 3 days, so you could have the time. Notice that they'll also incubate it at the same temperature, so temperature of the agar is another excellent control variable. But given what I've seen here, some other things which they've seen is they've used the same size paper discs, they've used the same concentration of each antibiotic. Any of those 3, excellent control variables. So I'm going to say they use the same concentration of antibiotic, and that again makes it a fair test, doesn't it? Okay, because I'm not favouring one antibiotic by giving it more concentration. Suggests why one disc was soaked in water. Now this is a source of confusion because you've got control variables, which are things within an experiment which you keep the same, but you also have something what we call a control. Okay, now what is a control in this sense rather than a control variable? It's something where I can compare to it. So by having a paper disc soaked in water, I would see the effect of no antibiotic. So I can see how well A, B, C and D do against just having a paper disc in the water. Okay, so the reason I have that control is to have something to compare to. Okay, so this acts as a control to compare to. We can see the effect of no antibiotic, because it could so happen that paper happens to naturally be antibiotic, and just by having a paper disc in the agar I kill some bacteria, but by having this paper disc soaked in water in there instead, I can prove, because there's no area around it of kill bacteria, that the paper itself is not responsible. Which antibiotic in figure 3 would be the best to treat gonorrhea? So I'm looking for the biggest area around it which is killed, because the antibiotic will spread out, and the larger the area around the disc, that means that kind of the lower concentration of antibiotic is enough to kill, because the antibiotic has spread out further. So here there's not going to be as much antibiotic as right in the disc. So here is still enough to kill the antibiotic. Basically, the bigger the area, the better the antibiotic. So the antibiotic I'm going to choose is A, because it had the largest area of bacteria killed. Basically killed the most, yeah? Okay, moving on to question 2. This question is about photosynthesis. What are two products of photosynthesis? So products, remember, are what are on what we call the right-hand side of the equation. They are things that are made. Yeah, they are products. When we think about products on the shelves, they are things which have been made. So in any chemical reaction, you have reactants and they turn into the products. So we're thinking about what is made. And the answer here is glucose is made, and oxygen. We often talk about plants giving out oxygen. They have made oxygen. They've also made glucose, which hopefully you'll recognise as the sugar which is made. You'll sometimes maybe have learnt it as sugar. So reactants, the actual reactants here are carbon dioxide and water. That's what the plant takes in, and the products, what it gives out, are oxygen and glucose. The student investigated the effect of light intensity on the rate of photosynthesis. Figure 4 shows the apparatus, and you've got an LED light source, you've got a test tube with pondweed in sodium hydrogen carbonate solution. You'll probably remember this from class, as if you've got dodgy pondweed, it almost never works, and your teacher will have made you sit there and watch a plant not bubble. You can see here there's a couple of bubbles coming off, and those bubbles are of oxygen, because that has been given out from photosynthesis. So they've placed it at 5 centimetres, they measure the rate by counting the number of bubbles in 30 seconds. They then repeat it at different distances, they'll move it further away. How could the student measure the rate of photosynthesis more accurately? Okay, so I've got different options here. So they could count the number of bubbles produced in 1 minute. They can measure the change in mass of the pondweed in 30 seconds. They can measure the volume of gas produced in 30 seconds. They could place the pondweed further from the source. They could use water instead of sodium hydrogen carbonate. So the sodium hydrogen carbonate is there as a source of carbon dioxide, because you can see here the carbonate. So when it dissolves, you've kind of got these this available carbon dioxide you can think of. So it's not going to be this one. And they place it further from the light source, then that is that's actually what we're changing in the experiment. So that's not going to help us measure it more accurately. The two ways which are going to be more accurate is measuring the volume, because bubbles are a bit rough, aren't they? Okay, because you know you'll have some small bubbles, some big bubbles. So if I measure the volume, if I put a gas syringe on there or something like that, then I've got a more precise and accurate measure. So definitely that one. thinking about the mass of the pondweed, obviously you're not actually weighing the pondweed, so I actually don't like this answer anymore. I actually think that if you're counting the bubbles in 1 minute, then you've got a more representative sample, okay? Because if you're counting it over a longer period of time, then the rate is able to be more steady, okay? If it's more up and down like this, and I only count for a little bit of time, I might get a high spike, okay, where overall I'll get a kind of a much larger picture. So I'm going to correct myself, and I'm going to say that number of bubbles in 1 minute is a better answer. The LED light source does not get hot. Why is this important? Okay, if I'm what I'm looking at here is I'm looking at the effect of light intensity. Okay, if that LED light source is getting hot, then that means that the temperature is also going to change. So it's important in any experiment that you're only changing one variable. Okay, so if the LED gets hot, the temperature will change. Okay. So temperature is what we call a control variable. We've discussed them in question 1. And it would affect the experiment, or affect the results. We only want to change light intensity. Okay, so we've got our results here. Distance of light source from pondweed in centimetres, and I can see here the bubbles. And just before I'm even moving on, okay, I'm seeing that as the distance goes up, the number of bubbles goes down. It should hopefully be what we expect, because there's going to be less light. Calculate the number of bubbles produced in 2 minutes when a light source was 10 centimetres from the pondweed. Now this is quite sneaky, isn't it? Because it's 2 minutes. Now if you remember back from our method, okay, we are measuring 30 seconds. So what that means we're after is, or here it says 30 seconds. So I want to look at my table for the result for 10 centimetres, which is 13. Okay, so 13 bubbles in 10 seconds, in 30 seconds, sorry. But remember we've got 2 minutes, so I'm going to have to times that by 4. So it's going to be 4 times 13, okay, which is 52 bubbles. Okay, a little bit sneaky, that one. Plot the data on table 1, draw a line of best fit. Right, first of all, whenever you see a line of best fit, you know that it is going to be a line graph. You also know this because both of the sets of data are numbers. Okay, so both numbers, if they're both in numbers, then that means line, or scatter as Excel calls it. If I look at the bar chart from earlier, okay, these are categories. Okay, I know it's a bit annoying because years are numbers, but if I had a line graph where I had red or blue, I can't really do a scale for that. Okay, I just have to have them in categories. So that's where bar charts come in. The second thing, and this contradicts I think with what a lot of you learn in maths, but a line of best fit is the line which best fits it. A line can be straight, okay, but it can also be a curve, and that's super important, and it's really hard to usually get the head around that, because I know maths will say lines best fit will always be a straight line, and then you turn up in science and scientists will say they can be curves as well. You're going to have to kind of just adapt to each lesson, but in science they can definitely be curves. They after all are the line which best fits the data. Okay, so let's plot it. So 5 centimetres is 40 bubbles, and I can see that they, if I read up from 5 and across from 40, they meet there. Okay, At 10 it's 13, so across here, and the scale's really nice, so each little square is 1, so I've got to 10, 11, 12, 13, 15 is 5, 20 is 2, 1, 2, 25 is 1, and 30 is 0. And that, it can also be a curve. It's super important now, because if I was trying to draw a straight line here, do I do something like this? And it's going to miss most of the points. If we actually have a look, the relationship is this kind of inverse square relationship. That basically means that it comes zipping down and then flattens out. Zipping is a technical term, by the way. It's not. Now, the easiest way to do this is if you've got a bendy ruler, you can bend the ruler. Or actually, the human eye is actually surprisingly good at it. So don't feather, don't like kind of do this weird sketchy thing. Okay, you just got to commit and go for it. Okay, so I'm going to turn it like this so I can kind of move my hand like this. Okay, and I'm just going to go for it. Look at that, beautiful. Now that it should be done in pencil. Okay, I'm just doing the whole exam in this one pen, but crack out a pencil. Give one conclusion that can be made from these results. Well, we already saw it without even looking at the graph. Okay, we said that as the light intensity, or as the light intensity decreases, or I suppose actually to make it less confusing, we could say distance from source increases, because obviously if you think about it, if I'm getting further away, the intensity of the light is going down. The rate of photosynthesis decreases, and that's because the number of bubbles goes down, because if there's doing more photosynthesis, that means there's more bubbles. Okay, because remember the bubbles come from photosynthesis. That's because the number of bubbles decreases. Now that's probably a bit confusing because I've tried to kind of be too fancy with it. If you just said as the distance to the light source increases, number of bubbles decreases, that's fine. Okay, but I've tried to kind of take it a little bit further. Question 3. The student affected the effect of different concentrations of sugar solution on pieces of potato. Okay, so this is our osmosis practical, our required practical that we've looked at. This is the method used. Cut 3 pieces of potato to the same length. Dry each piece of potato on a paper towel. Weigh each piece. Place each piece in a different concentration of sugar solution. Leave all 3 pieces for 2 hours. Remove the 3 pieces of potato from the solutions. Dry each piece on the paper towel. Measure the length and mass of each piece of potato, and you can see them here. Why does the student dry each piece of potato before weighing it? Okay, so if I take something out of water, it's wet, right? And water has got some mass. So if I'm about to weigh it, I want to weigh the potato, not the weight of the water on the potato. So by drying it, I'm going to remove the water and therefore get a more accurate measurement. Okay, so to remove excess water which would increase the mass. Okay, we want to weigh just the potato. What 2 changes would you expect in the potato in tube A after 2 hours? Okay, so this water is just pure water, there's no sugar there. Okay, so if I think about what osmosis is, okay, it is the movement of water from high concentration to low concentration across a partially permeable membrane. Okay, I'm not going to write partially permeable membrane. The potato itself, okay, is a partially permeable membrane. Okay, now I always find osmosis really confusing because it's the movement of water from high to low. But if I'm looking at the sugar, right, remember the sugar doesn't move. Okay, the sugar doesn't move, but the mass will move from where there's low sugar to high sugar. Because if I'm focusing on the water, that's high water to low water. Let me try and explain that in a different way. If I here, okay, have a glass of very sugary solution, so these big circles are the sugar, so that's high sugar, and the dots are the water, so that's high sugar but low water. And if I get this here, okay, if I have just say 1 sugar and the rest is water, okay, then that's low sugar and high water. So when it says osmosis is the movement of water from a high concentration to low concentration, here there's lots of dots, okay, so high concentration of water. Here there's not so many dots, so it's a low concentration of water, so it would move this way. It would spread the water, the dots would move this way. But if I'm looking at the sugariness of the solution, it's gone from low sugar to high sugar. Those big dots don't move, it's only the little dots, okay, which can move. Okay, the water. So in the potato, okay, I'm going to have water. I'm also going to have the other particles which are dissolved in it, which make a potato. And outside, I've just got water. I've just got those dots. So though that water is going to move in, because it's from high water to low water, and that means that the potato is going to swell and it's going to weigh more. So that means I'm going to have an increase in mass and an increase in length. Okay, the potato will get bigger and heavier because the water from outside has moved in. Complete the sentences. Now look, I've in explaining that, I've already leapt ahead. Water moves into and out of cells by a process called osmosis. Water would move into the potato cells in tube A. The solution outside the potato in tube A is at a higher water concentration than the solution inside. Now, I actually don't like this question. I think this is a very tricky question. I've put higher water concentration just to make sure. Okay, I don't like this question at all because you know, osmosis is confusing because you've got to think about, are you looking at water, are you looking at sugar? So I've made sure by putting higher water. I might have a very pernickety examiner who might dock the mark, who knows. The potato in tube B did not change. Give one conclusion that can be made from the observation. So if the mass doesn't change, that means that the concentration is the same outside as inside, okay, because it will move in as fast as it moves out. Okay, it's at what we call equilibrium. It means that everything's balanced. So what conclusion can be made? Okay, that means that the potato is at the same concentration as the solution. Now this is a very fancy word, but actually you'll know this from looking at like Lucozade and energy drinks. It's what we call isotonic. So when you get an isotonic sports drink, the reason that's so useful is because it's at the same concentration as your body cells, so it means that you can kind of replenish it. Unless you're running a marathon, just drink water, seriously. So, moving on. Okay, figure 7 shows the root of a germinating seed. Describe 2 ways the root is adapted to absorb water efficiently. Okay, look at all these hairs like this. Okay, that means it's got a superb surface area, and surface area is really important for osmosis. If I have a larger surface area, it means it will happen faster. I like to think about this as, imagine you've got to get people into a hall, right? If I have one door for everyone to get through, so here's all the people and they want to get through. If I have one door for them to go through, it's going to take ages. They have to line up and everything like that. If I have loads of doors, double doors, all this type of thing, everyone can rush through into the room. Okay, I've got more places to get in, and if I have a higher surface area, okay, that means I've got more places to get in. So I've got these root hairs. to increase the surface area. Okay, it's hard to see on the copy. It's white. I mean, that's the main one. Okay, it's hard because of the fact that it's a germinating seed. You haven't really got like leaves where you've got this transpiration stream to reduce the water concentration. Okay, so as well as the increased surface area, and you've got these root hairs, they extend into the soil and have really thin walls. If I have a very thin wall, that means it can osmosis can happen much more quickly. So I'm going to say that the root hairs have thin walls. So, question 4. Exercise can improve health. A student measured her breathing rate at rest, when walking, when jogging. Figure 8 shows her results, and you can see here. Compare the breathing rates when doing the 3 different activities. Use values from figure 8 in your answer. So whenever you see use values, you've got to be able to read the graph. So again, I'm going to use my ruler. Okay, measuring across, I'm 1 little square up. Now, because there's 5 little squares between 10 and 20, that means each little square is 2. So that means at rest, we are looking at 12. Walking, 2 little squares up from 20, so that's going to be 24. And jogging, I'm sure you can probably already see, but that is 60. So we know that that's 60. Okay. So it says compare, so that means I've got to just look at them, comparing them to each other. I don't have to do actually any explanation, but it might help your answer if you just start to give a little bit of explanation. So at rest, the breathing rate was lowest. At 12 breaths per minute. Okay, this increased with greater activity, because obviously walking is more activity than just sitting around, and jogging is more activity than walking. So walking, so that's I'm going to say a mild increase in activity, or mild increase in effort. It doubles, because 24 is twice 12. Whereas for jogging, it increased 5 times to 60. Now, that doubling and increasing by 5 times makes it really clear that I'm comparing results, okay? Because the only way that I can tell that it's doubled or quintupled is by comparing it to the other one. Explain why breathing rate changes when doing different activities. So this is all going to come down to respiration. Okay, now this is kind of the opposite of photosynthesis. So respiration is glucose plus oxygen goes to carbon dioxide, excuse the poor equation form, plus water plus energy. I'm going to put that in brackets because it's not a substance per se, plus energy, and that's why we're doing it. We want energy, so we're using our food, we're using oxygen from the air, and we're creating energy that we can use to move and do all the functions that we need. Okay, and doing those different activities, we need more energy to move. Okay, so during activities, we need more energy for movement. Okay, that's my first point. To get this energy, our bodies use respiration, which requires oxygen. Okay, so a greater breathing rate means more oxygen, and therefore more respiration and more energy for movement. So notice how I've done kind of a logical chain there. I said that this is this, therefore this. So figure 9 shows the heart and circulatory system, and it says a heart is a double pump. Describe what this means to you. Use figure 9 to help you. Okay, hopefully you can see that there's 2 loops here. Yeah, one to the lungs, okay, and one to the body. Okay. So the reason it's a double pump, okay, is because there are 2 circulatory loops. Okay, and I'm going to embellish it with facts from here. Okay, because you can see that the left ventricle pumps out, so it actually goes this way like this to the body. The right ventricle pumps to the lungs. The left ventricle pumps blood to the body, and the right to the lungs. Okay, the wall of the left ventricle is much thicker than the wall of the right ventricle. That's just one reason for this. Now, first of all, I haven't mentioned this yet, but these diagrams are quite confusing because if you think about it, the left ventricle is on the right of the diagram, and the right ventricle is on the left. Now, this is quite historical because it comes from when people would dissect, okay, and they'd cut open the body, and obviously when you're looking at the body, you're looking at, you know, from the top. And if I lifted up this exam paper and put it on my chest, you'd see that then the left ventricle would be on the left half of my body. Okay, so even though it's the left ventricle, when you look at it in a diagram, it's on the right, because that's what you'd look at if you were cutting up a body. Morbid, I know. So the wall of the left ventricle is much thicker because it's got to go to the whole body. Okay, and the lungs are right next to the heart, so it only needs to kind of thud just over to the lungs, whereas the left ventricle has to pump blood to every single cell in your body. So that's going to require much more oomph, much more muscular power, and is therefore going to be a thicker wall. Okay, so the left ventricle pumps blood to the whole body, so requires thicker muscle and more pressure to do this. Okay, people encouraged to exercise after recovering from a heart attack. It's just one reason why. Exercise is really beneficial because it makes your body more efficient at moving oxygen and other substances around the body, and it helps them get used. Helps your body get used to higher and more stressful situations. So exercise is going to be really useful after a heart attack because then your body can the heart can strengthen, you can improve that circulation and reduce and control your weight, which is probably a factor in your heart attack in the first place. So by exercising, okay, you can strengthen the heart muscle. It's a muscle, right? And when you exercise like your biceps get stronger. You can strengthen the heart muscle and improve circulation. My handwriting's going a bit rubbish here. Apologies for that. Okay, question 5. Cells divide into a series of stages called the cell cycle. Stage 2 of the cycle is mitosis. So figure 10 shows a simplified cell cycle for a human body cell. So you've got stage 1, stage 2 mitosis, stage 3, and then background. Okay, so you're going round and round and round. Draw one line from each stage in the cell cycle to what happens during that stage. So mitosis is is how your body makes new identical cells and it does this for growth and repair. Okay, so if I asked you to make to take this, after I've done this, to take this exam paper and make a whole new one, you would, and I tore up all the pages, so it's just page after page, and I asked you to do this, you'd probably come up with mitosis. So what the first thing you'd do is you'd go to the printer, the photocopier, and you'd copy every single page in this exam. You'd then take them and sort them out, okay, in each pile, so you know you've got, so I'd take page 1, page 1, page 2, page 2, page 3, page 3, page 4, page 4, and you'd sort them all out, and then you'd separate them out into the two piles. And this is exactly what our cells do. So our cells start off like this with your nucleus. I'm just going to draw 4 chromosomes, for instance. I'm then going to kind of replicate all those. 1, 2, 3, 4, 5, 6, 7, 8. So I've got 8. The nucleus is going to kind of disappear, and those copies are going to line up one against each other down the middle. I get these things called nuclear spindles, which act like string, like this. They're then going to pull apart, like this. 1, 2, 3, 4. Okay, and then finally you can see that I've drawn the cell membrane kind of snipping in. I'm going to get 2 identical cells with 4 chromosomes. Okay, so the stages are replicate, line up, pull apart, and divide. Okay, so stage 1 here is the first thing, which is going to be the copies of the DNA are made. Okay, stage 2 is this nucleus divides. It's going to kind of break up and line up, and stage 3 is the dividing into 2. Looking at question 5.2, it says the mass of DNA in the human body at the start of the cell cycle is 6 picograms. Okay, now you don't actually need to worry about picograms, just the number is what's important here. What mass of DNA will be in each of the new cells produced by this cell division? Now here's the thing, because I'm making identical cells, that means that whatever I start with, I'm going to have, if I had 6 picograms here, okay, I've doubled it, so that's now 12, so I've still got 12 here, 12 in this whole thing, and it's going to split back to 6. Okay, I've got the same at the start as I have at the end, so it's going to be 6 picograms. The next one says stem cells are undifferentiated cells. What statement about stem cells is correct? Animal stem cells are found in meristems. Meristem is in plants. And also animal stem cells divide by meiosis. They don't. That is only the gametes. So that is egg and sperm. Meristem cells in plants can differentiate through the life of the plant. Okay, or that meristem cells can differentiate into only one type of cell. Meristem cells in plants can actually differentiate throughout the life. Okay, this is a little bit different to humans. If I take, might be able to know this because you can take cuttings of plants and put them in the soil, and it will grow new roots. It will grow stem, whatever, and actually make a whole new plant. So that tells me that those meristem cells can make any type of plant cell. So that is the answer there. Stem cells from human embryos can differentiate into most types of human cell. Research is being done into the use of embryos of embryonic stem cells in medical treatments. The long-term effects of using embryonic stem cells in patients are not well understood. In therapeutic cloning, human embryos are produced using donated human egg cell and a cell from a patient. The embryo produced contains the same genetic information as the patient. The stem cells are taken from the embryo and stimulated to divide to form the cells the patient needs. The embryo is then destroyed. It's just two advantages of therapeutic cloning. Okay, it's quite a lot here. First of all, you're using the patient's own cells, so that means that they're probably not going to be rejected by the body. So one of the big problems with organ transplants is if I, say, received a kidney from another person, my body detects that kidney is not mine and will start to reject it and kill it because it thinks it's a threat to the body. And you need to take immunosuppressant drugs which kind of stop your immune system from doing this. So there won't be rejection. Okay, so you've got a low risk of rejection. Also, my early dating life, I wish. Number two, first of all, if it's an embryonic stem cell, you can make most types of human cell. So I could, if I had, for instance, a problem with brain or a problem with heart, I can make new of those cells to do it. Okay, so I can make a large variety of cells to cure a large range of diseases. For instance, I could produce replacement organs. I could potentially make a whole new kidney from my own cells, and then I wouldn't need to undergo a kidney transplant. It's just two disadvantages. Okay, so this really comes down to the ethics. Okay, to start off with, the first thing is with embryos is that the embryo is then destroyed. So because it's an embryo, in theory, okay, you could implant it into a mother, and that could grow and develop a baby. That's not always the case, but we're going to say it is. Okay, so by destroying embryo, means you have destroyed a potential life. Another thing is you have got to donate the human egg cells. Now that's going to be quite difficult. It's not actually the nicest process. It involves quite a large needle and some fertility drugs to start off with. So you're going to have to convince people to do this. This egg donation has risks. Okay, and there might be a shortage. Some other points that you could have made is to say that this kind of stimulation is quite hard to do, so you have a poor success rate. And also, this is untested. Okay, research is being done. It means it hasn't been done. So that means that you don't know the effects in the long term. So moving on to question 6 then. It says cell structures, draw one line from each cell structure to the type of cell where the structure is found. Okay, so nucleus is eukaryotic cells. Eukaryotic cells have nuclei, and what we call membrane-bound organelles. Now, that's a fancy word, but nucleus is a membrane-bound organelle. But that basically means mitochondria. That means vacuoles, and that means chloroplast. So permanent vacuole has got to be in eukaryotic cell, but a permanent vacuole is actually only in plant cells, and plant cells are eukaryotic cells. Plasmids, I like to think about them as DLC for games. Okay, so obviously you might be familiar with this, but you buy a game. So here's my game, and then you play the game, and then they might release a little extra add-on pack, DLC, downloadable content, where you pay a little bit extra money and you get a little bit more game. So in bacteria, you have this chromosomal DNA. That's the main kind of DNA of the bacteria, and then you can have these little extra bits of DNA which can add on little extra genes which maybe could carry antibiotic resistance and other things which are useful to the bacteria. So like a game and its DLC. So plasmid is only in prokaryotic cells, and bacteria are your big example of these. So they don't have a nucleus. Notice how I drew that as a loop of DNA, not a nucleus. Okay, that is the DNA floating about on its own. So that's bacteria and stuff like that. Moving on. Figure 11 shows a plant cell. What are the names of structures A, B and C? So A is pointing to the vacuole. So I know that it can't be this row because it hasn't got that there. And now I've got to decide here. Now C is pointing to the right outside. So this is a plant cell. So the right outside of a plant cell is the cell wall, not the cell membrane. But just to clarify, we'll just make sure of this. B is pointing to the smallest structure I can see here. And the smallest structure in a plant cell, or most eukaryotic cells, is a ribosome. They're very, very tiny. Can't see them with a light microscope, need an electron microscope. So that means the bottom row is the one that we're after. The student observes slides of onion cells using a microscope. Figure 12 shows 2 of the slides the student observed. Here it's a little bit fuzzy, but you can see they're much bigger than they here in slide B. The cells on the slides are not clear to see. Describe how the student should adjust the microscope to see the cells on slide A more clearly. So hopefully this is really asking you about microscope technique, and asking you how do you focus a microscope. And there is hopefully you'll remember that there's a kind of a focusing knob that you can turn, and that will adjust the height and therefore focus it. Because this is so zoomed in, I don't think it needs that much. So I'm going to say you could turn the fine focusing knob, because there's a coarse one where it does it really quickly, and there's a fine one where it's a little bit more slowly. Okay, until the cells are in focus. Because, I don't want to go past that. The cells are in focus. Okay, 6.4, describe how the student should adjust the microscope to see the cells on slide B more clearly. So this is in focus, but you can see it's not as zoomed in as this. So if I was wanting to see individual cells more clearly, I would switch to a higher power. Now, if I'm looking at a microscope, you have 2 lenses. You have the eyepiece lens, is what you look through, and then you will tend to have this kind of thing here where I've got kind of 3 options, and you're only really looking through one. Okay, at my slide like this. This is the worst drawing of a microscope ever. You can tell I didn't do art GCSE. So this one might be 4 times, this one might be 10 times, this one might be 40 times, or something like this. So what I can do is I can rotate this and change to a higher power. So I would change the objective, so this is these lenses here are called the objective lens, to a higher power. Okay, continuing on. Lovely. 3 pages left. Time flies when you're having fun, you know, because this is what I like to do in my free time. He says, the student made the necessary adjustments to get a clear image. Figure 13 shows the student's drawing of one of the cells. The real length of the cell was 280 micrometers. Calculate the magnification of the drawing. So, this is now we're in the questions which appear both on the foundation paper and the higher paper. So this is where they might now start to ask you things where you have to remember them a little bit more. So there's only really one major equation that we are faced with in biology, and that's the magnification equation. So that states that magnification equals image size divided by real size. Okay, so I know the real length is 280 micrometers. Okay, that micro is going to be important because this only works if they are in the same units. And I don't even need to get my measurement out, but actually, I just fancy checking the exam, see if they're right. I tell you what, there you go, 112 millimeters. Look at that, beautiful. So I'm going to put it into this equation, but the first thing I'm going to have to do is change either the micrometers into millimeters or the millimeters into micrometers. I'm going to change the millimeters into micrometers. Now, unit conversion is one of the hardest things that you have to deal with, really, and it relies on us knowing the unit system. Okay. In the metric system, we realised that it's much easier to work in tens, okay, or thousands, where it's a nice round number and the units scale really nicely. Back in the old days when you had imperial, and obviously some of you'll still be familiar slightly with imperial, there are, for instance, 3 feet in a yard. There are 1,760 yards in a mile. But you could also have 8 furlongs in a mile. It gets really weird, okay, and the units get really bizarre. So what they said is we'll make everything on nice powers of 10 or /1000, okay. So what we're going to do is we're going to take the unit meters, because that's the unit for length. And if I'm dealing with smaller lengths, I'm going to add something at the front, okay, it tells me I'm dealing with smaller lengths. So I'm going to add another m. And it doesn't actually matter what the units are. If I add a little m at the start, so if I had litres, for instance, and I had ml, that's millilitres. If I had grams, g, it'd be milligrams. And what that little m at the start tells me is that it is 1,000 times smaller. Okay. If I want to go 1,000 times smaller still, now this unit is a bit weird. That is the Greek letter mu, and that's the Greek letter m, so it's like an even smaller number. And that is 1,000 times smaller still. Okay. So, if I've got 280 micrometers, right, so I've got 280 of these, and I want to convert it up to millimeters, that means that because each one of these is 1,000 times smaller, that means that I'm going to have to divide by 1,000. So 280 micrometers is going to be 280 divided by 1,000, which is 0.280, I suppose, millimeters. So now I've converted it, I can put it into the equation. So magnification equals our image size, so that's the size of this image, what I've just converted. I haven't just converted it, it's already given to me in 112 millimeters, divided by my real size, which is what I have converted over, and is 0.280. And I can put that into the calculator, use a calculator. Remember, there's no non-calculator science exams. I'm going to use Siri again. Hey Siri, what's 112 divided by 0.280?
112 divided by 0.28 is 400.
Thank you. And that means that my image has been magnified 400 times. Okay, good rule of thumb is your magnification is always going to be above 1. If it's smaller than 1, the question has been very mean, but if the magnification is smaller than 1, the chances are you've probably got the numbers the wrong way round, and you've put the real size at the top and the image size at the bottom. Okay, last question. Coronary heart disease is a non-communicable disease. Coronary heart disease is caused when fatty material builds up in coronary arteries. Explain what a non-communicable disease is. So if you look at the word communicable, that to me shouts communicate. Right? And when you communicate, that means that you are spreading information. You know, if I'm talking or communicating with someone, I'm spreading the information of what I'm saying to that other person. So if I've got a non-communicable disease, that means that I can't communicate it, I can't spread it. Okay, so a non-communicable disease is one which cannot be passed on. And why can't it be passed on? Well, that's because it's not caused by a pathogen. Remember earlier we discussed the pathogens and we said about bacteria, virus, fungi and protists. They are little things which can be passed on. Okay, so if it can't be passed on, it means it's probably not going to be caused by a pathogen. So non-communicable disease is one which cannot be passed on. This is because it is not caused by a pathogen. Okay, figure 14 shows a coronary artery of someone with coronary heart disease. And you see the artery wall here and the fatty material built up. Okay, so explain how that can cause a heart attack. So if we're looking at the diagram, you can see here that that space is much smaller because of that fatty buildup. So if I'm restricting that flow of blood, that means that I'm going to get less oxygen and other nutrients to the heart, and that means that the heart muscle, which has always got to work, okay, is going to get less oxygen to it, so it means it cannot respire, do that respiration to get that energy to contract. Can't do that as much as it needs to, and that means that heart muscle will start to die, and that is what we cause a heart attack. So coronary heart disease reduces blood flow through the artery. Okay, this means that less oxygen can get to the heart muscle. This means not enough oxygen for respiration, and the heart muscle will begin to die. And that's what a heart attack is, where part of your heart has started to die. The last question, it's a nice 6-mark question to finish. What a treat. It says, explain how lifestyle and medical risk factors increase the chance of developing coronary heart disease. Okay, so this is a long answer question, so I'm going to use Rustman to answer it. Okay, remember these are purely advisory, it's not like a step-by-step process. It is in some regards, but it's a little bit more vague than than a prescribed plan. So R is read the question. So I'm going to see here, it says explain how lifestyle medical risk factors increase the chance of developing coronary heart disease. Underline the command word. So I'm going to look at explain. That means I need to say why those things are important. Subject words in here. We've got lifestyle factors, we've got medical factors, and coronary heart disease. Think of any other keywords. I'm thinking straight away things like obesity. I'm thinking things like diabetes, exercise, alcohol, things like genetics. These are some other keywords that maybe I'm going to want to mention. Make sure you organise your answer. So I'm going to take it in 2 paragraphs, and I'm going to look at the lifestyle factors and the medical factors, answering all the question. Again, this is really important in a question like this, because if you choose only lifestyle factors and only medical risk factors, then you're excluding one half of the answer, and that means you're not going to be able to get all the marks available. And then once we're done, we're going to go back and check. So lifestyle factors, okay, include things like smoking, because that's a choice, isn't it? Okay, you are choosing to smoke. Things like obesity, it's a little bit more vague, but obviously if you eat too much, then that's not going to help. Things like lack of exercise, things like a high fat and salt diet, and one more might be air pollution. So for instance, you get more coronary heart disease in urban areas. So medical factors, so I'm going to start a whole new paragraph, so I'm organising my answer, include things like high blood pressure. You might not be able to help that. That's a medical thing, isn't it? You don't go, I'm going to have high blood pressure today. High cholesterol, things like diabetes, as we said, and things like and genetics. Okay, you don't choose the genetics that you're born with. Notice how, if I stopped here and I read back, I'd notice that I haven't actually answered the question, because I've just stated those factors, I haven't explained. So I'm going to add one more paragraph just to explain how they actually increase the chance. So obesity and high cholesterol. Notice I'm picking one from each there. Can cause fatty deposits, those things that we saw on the previous question, building up in the arteries, and coronary heart disease. Things like smoking and high blood pressure. So smoking, and actually we're going to go with high salt diet can cause high blood pressure. And if you think about it, if I'm really pumping the blood through that small cavity, then that means that some of this fatty material might break off and actually cause a heart attack, which increases stress on fatty deposits, leading to heart attacks. And one more is I'm talking about the genetics. Okay, genetics can affect how easily you develop coronary heart disease. Okay, that's not the best answer, but I think I've hit all of the points. So just reading back, lifestyle factors include smoking, obesity, lack of exercise, high fat and salt diet, and air pollution. Medical factors include high blood pressure, high cholesterol, diabetes, and genetics. Obesity and high cholesterol can cause fatty deposits and coronary heart disease. Smoking and high salt diet can cause high blood pressure, which increases stress on fatty deposits, leading to heart attacks. Genetics can affect how easily you develop coronary heart disease. Again, it's not the best answer. If I had a mean examiner, I probably might not even get full marks, because it's weird. These 6-mark questions are very weird to mark. But notice by following that, I've increased my chances of getting those high marks by making sure that I organise my answer, making sure I'm including keywords, and making sure I'm answering all the questions. So that is the 2018 foundation paper. I hope you found that useful and not too boring.